components on the market would be to perhaps pre-vascularize the scaffold using autologous derived stem and progenitor cells. This is put differently pictorially. You can see on your left-hand side an existing dermal scaffold with not particularly good graft take, and by perhaps implanting your scaffold with stem cells or pre-vascularizing the scaffold, you'll hopefully result in a better functional and cosmetic outcome. In order to produce this pre-vascularized scaffold, you need three components. You need the matrix itself, and we know that skin is mainly composed of type 3 and type 1 collagen, elastin, some glycosaminoglycans, including hyaluronic acid. You also need to produce your tubules endothelial cells, and to support them stromal cells. And ideally these cells should be from an easily isolated and easily expandable population, such as stem and progenitor cells. So the endothelial cells I used were called endothelial colony forming cells, and they were first identified in the late 1990s. They were called EPCs at this stage, and they were thought to be endothelial progenitors, but in fact contained other markers, HSC markers, and actually when cultured in isolation did not form vascular tubules. Further investigation of these cells, and in particular taking a CD40 negative fraction of them, identified another cell line which formed colonies in vitro, and these were called ECFCs, or endothelial colony forming cells. They had a cobblestone morphology in vitro, and they were shown to be able to be isolated from HUVECs, umbilical vein endothelial cells, cord blood, and more recently we've been able to isolate them from peripheral blood and even adipose tissue. Myself, I isolated the peripheral blood ECFCs from something called a leukocyte cone. So I worked in a blood donation centre, and blood is normally leukophoresed because the white cells are not used in the blood transfusion to try to protect the recipient of the transfusion from transmissible diseases. So these white cells would normally be thrown away, but instead the leukocyte cone contains the mononuclear fraction, and this can be plated in endothelial growth media and forms the classic cobblestone morphology, with positive endothelial cell markers and negative for mesenchymal and hemopoietic cell markers. And this is shown by the characterization, which shows it's positive for the sort of 31, 144, 166, 73 endothelial markers, and negative for things like CD14 and CD45, which are hemopoietic cell markers. In terms of isolating the adipose-derived MSCs, MSCs, as I said, are required to stabilise the newly formed tubules in my scaffolds, and they've been shown to be isolated from multiple cell tissues within the host, including bone marrow, but more recently adipose tissue. And obviously adipose tissue is much more palatable for a patient. So if a patient comes into the hospital, it's much easier to isolate them from a lipoaspirate than a more invasive isolation from bone marrow. So in my experimentation, I used fat that would otherwise again be discarded. So breast reconstruction patients, we don't use the whole of the abdominal pannus for reconstruction of the breast. We just use the central portion, and normally the zone, what we call the zone 4, the part with the least vasculature, is thrown away. So this can easily be used for experimentation purposes. And from this, you take the fat, mince it, and then digest it with collagenase. Take the stromal vascular pellet and this is what I did, and again the markers were consistent with a mesenchymal cell line, had negative markers for endothelial cell lines, and on trilineage differentiation confirmed they were indeed adipose-derived MSCs. Having isolated my ECFCs and adipose-derived MSCs, the next course of action was to try and find the best scaffold to implant them into, and I used a proprietary device called the BD mu chemotaxis model, where essentially one places your scaffold choice. So in this case I used mainly collagen and fibrin, as collagen is the main component of skin and fibrin is the main component of the fibrin clot with wound repair. You place that in the central column with your cells of interest, and on either side there's a reservoir into which you place a chemoattractant to try and get your cells to move. I can play the video whilst I'm talking so you can see what I mean. And it's quite hard to see, but essentially over a 24-hour period of time lapse, you follow your cells, you track the cells over time, and it gives you a lot of information in terms of how far they move, how quickly they move, and that gives you an idea as to what would be the best thing to build your scaffold with. So I tried different collagen and fibrin gel concentrations. I used 3 endothelial colony-forming cell lines, HUVECs, cord blood ECFCs, and peripheral blood ECFCs. And in some of the gels I also added fibronectin, as this often helps with cell migration. This is the proof of concept for the device, and you can see that it gives you information. This is 30 cells that have been tracked using the cell time-lapse and program called ImageJ. You can see when there's no chemoattractant, so 10% FCS on both sides, the cells migrate in a rather haphazard manner, but when you place a 10% gradient onto one side, the cells migrate towards that gradient. These are some of the results. Obviously I haven't got time to go through all of them, but you can see just from these that the peripheral blood-derived ECFCs with fibronectin, this is a 2 milligram per mil collagen gel, migrated further than any of the other cell lines, the HUVEX or the cord blood-derived ECFCs, and they migrated towards the 10% FCS gradient. So this is when there's no gradient, so 10% FCS on both sides, and this is when there's a 10% FCS gradient, the cells migrate towards that. So the device is useful for determining which is the best scaffold, but it could also be useful if you wanted to add something else to the reservoir, something like VEGF or something else that could attract to a cell to try and make a better scaffold. You could put that into the reservoir and get a better idea as to what would be the best thing to put into your scaffold. Having determined that a 2 milligram per mil collagen gel would be best, I then implanted my ECFCs and MSCs within this gel in a 96-well plate. I found that 200,000 endothelial cells to 30,000 MSCs was the best concentration. I added fibronectin to the gels because in the chemotaxis models this proved to increase the speed and the distance to which the cells travelled. And I compared bone marrow-derived MSCs to adipose-derived MSCs, and I compared the 3 different ECFC subtypes. These are some of the gels that I produce within the dishes. I then quantified the tubule networks that formed using confocal microscopy and a program called Imaris. And this gave me information in terms of the number of tubules that formed, subtubules, the total tubule area, volume, length, diameter, branch points, and gel thickness. And this is the Imaris device, if it will play. I don't think it wants to play, but it's fine. It basically just gives you this sort of information. These are some of the results where we're comparing bone marrow-derived MSCs to adipose-derived MSCs. These are EGFP-labeled ECFCs within the collagen 2 milligram per mil gel. And you can see that when you compare the bone marrow-derived MSCs to adipose MSCs, the tubules are very similar, whether you use HUVEX, cord blood ECFCs, or peripheral blood ECFCs. And that all 3 ECFC cell lines produce similar tubules, produce good tubules, which is useful from a clinical standpoint. Clearly, on a clinical basis, we can't use HUVEX or cord blood-derived ECFCs because of the immunogenetic potential. But if we can isolate the peripheral blood ECFCs from adult patients, we could use them and produce nice tubules in our scaffold and use them for a patient. These are some of the results graphically. And again, the number of tubules that formed was similar, as you could see from the photos, and there was no statistical significant difference between the subtypes investigated. But just taking, for example, adipose MSCs and peripheral blood ECFCs, there were significantly more tubules in this than adipose-derived MSCs and HUVEX. To summarise all of the results, as I said, there were more tubules when we co-cultured adult-derived adipose MSCs and peripheral blood ECFCs than any other MSC or endothelial cell co-culture, which is very encouraging. There were more subtubules with bone marrow MSCs than adipose MSCs, although this was not significant. Total tubule area and total tubule volume were higher when peripheral blood ECFCs were combined with bone marrow MSCs than adipose MSCs, although again this was not significant. And total tubule area and total tubule volume were actually higher when cord blood ECFCs or HUVEX were co-cultured with adipose MSCs than bone marrow MSCs. Moving on to some in vivo work, we used an immunodeficient murine model, and I upscaled my optimized gels from the previous experiments and compared those to an empty gel on the dorsum of these mice. The mice were sacrificed at day 14, and the perfusion of the scaffolds was examined. Here we can see some of the results, and you can see that in the control gel at day 14, There was no evidence of red blood cells within the collagen gel. But if you look at the prevascularized gel, there were numerous networks that had formed with red blood cells within them. And certainly per times 40 high power field, there were 21 tubules on average in the prevascularized mice and only 3.3 in the control gels. So the main conclusions were that adult-derived peripheral blood ECFCs and adipose MSCs provide an easily sourced and expandable progenitor cell population suitable for creating a microvasculature within a collagen scaffold in vitro. Future directions would be to carry out further in vitro experimentation, perhaps using these cells implanted within the gels before they've formed networks to see if that improves graft take. One could produce a bilayer prevascularized scaffold by seeding some epithelial progenitors onto the surface of these scaffolds. And one of the rate-limiting steps in this work is the fact that it takes peripheral blood ECFCs quite a long time to grow to a number that would be suitable for clinical applications. So it'd be nice to try and expand them more rapidly using bioreactors. I'd like to acknowledge my supervisors in Oxford and at UCL and the sponsors of my research, Restore Burn and Wound Research. Many thanks for listening.